Bismillahirrahmanirrahim. Assalamu alaikum class. Today we will discuss isoenzymes. The key concepts are: what are isoenzymes? Why there is existence of isoenzymes in a body? What is LDH isoenzymes? And what happens when there is change in levels or concentration of lactate dehydrogenase enzyme or LDH. First of all, what are the isoenzymes? The multiple form of an enzyme catalyzing the same reaction are isoenzymes or isozymes. It means that same type of reaction is catalyzed by multiple form of a same enzyme. Therefore, these multiple forms of enzymes are collectively called as iso means same, isoenzymes. But these forms of enzymes, they differ in their physical and chemical properties, and the important properties they are included as the structure. It means that their structure is different. They are made up of different structural entities. They have different electrophoretic mobilities, like in electrophoresis on the electrophoretic bed or the gel agarose, they will move to different extent. And immunological properties, Km, Vmax value, Michaelis-Menten constant, maximum velocity which can be attained, pH optimum levels, and their relative susceptibility towards the inhibitor. It means that which type of inhibitor is important for suppressing them or for causing their catalytic inhibitions, and their degree of denaturation, which means that these type of multiple forms of enzymes may be denatured to different extent when they are irradiated to different types of radiations, when they are under the effect of different pH or the temperatures. Next is why there is existence of isoenzymes. Therefore, explanation is needed for the existence of isoenzymes, and many possible reasons are offered to explain the presence of isoenzymes in the living systems. Okay, why these isoenzymes are present in the body. Number first is isoenzymes. They are synthesized from different genes like malate dehydrogenase of cytosol is different from that which is found in the mitochondria. It means that malate dehydrogenase enzyme is present in the cytosol and it's also present in the mitochondria, but they have the different, they differ in their properties. The main difference is that the different gene is responsible for encoding or for their synthesis. And second reason is that the oligomer enzymes, oligomer means the enzymes which are consisting of more than one type of subunits. It means that they are consisting of the subunits which are different in their properties. And the best example is for the lactate dehydrogenase and Critten Phosphokinase. And both enzymes, their functions, their levels, and the difference or the elevation in the level of certain isoenzymes have also been discussed in the upcoming slides. Therefore, these like lactate dehydrogenase and creatine phosphokinase, they consist of the different subunits. And next is an enzyme may be active as monomer or the oligomer. It means that in for the certain enzymes they may be activated, behaves as the catalytic entities or units when they are present in the monomeric form, but some are active when they are present in the oligomers, which consist of more than one subunits. And the example for such type of the isoenzymes are the glutamate dehydrogenase. Next is in the glycoprotein enzymes. It means that glycoprotein enzyme, they consist of the proteins, amino acid sequences along with certain carbohydrate units. Therefore, the name is called the glycoproteins. And the differences in the carbohydrate contents, it means that some isoenzyme unit may have a different carbohydrate content, but the protein content is same one. And therefore, due to the difference in the carbohydrate content, which is responsible for their presence or their action as the isoenzymes, like the alkaline phosphatase. And first is the best example for the isoenzymes and their functioning is the lactate dehydrogenase, which is LDH. And among the isoenzymes, LDH has been the most thoroughly investigated enzyme, which has been investigated by the multiple scientist group of analysts and researchers have undergone many researches on the plasma, serum, blood samples of different individuals, healthy and which are uh, the patients which have the multiple problems of the heart, muscles, and like that. And LDH, whose systematic name is L-lactate NAD nicotinamide dinucleotide oxidoreductase, which catalyze and enzyme commission like its numbering and its identity as 1.1.27, and it catalyzes the interconversion of lactate and pyruvate, these two uh, compounds. And therefore, here is the interconversion. Therefore, lactic acid can be converted into pyruvic acid, and similarly, pyruvic acid can be converted to lactic acid back by the action of LDH, but the presence of NAD is required. And therefore, when the lactic acid is going to be converted into pyruvic acid, it means that there is only one change, like hydroxyl OH group has been transformed into carbonyl group. It means that oxidation occurs. And oxidation occurs in the sense that hydrogen will be removed from the oxygen, and similarly, hydrogen will be removed from the carbon. And therefore, such carbon will be changed into carbonyl functionality, and therefore, two hydrogens are removed, and they appeared as like NAD oxidized has been converted into reduced NADH plus H positive. And overall, LDH has five distinct isoenzymes. These are recorded as LDH1, LDH2, 3, 4, and 5. These are the five distinct important isoenzymes of enzyme LDH. And these isoenzymes, they can be separated by the electrophoresis. And for the electrophoresis, I have told you earlier that like proteins or nucleic acid, DNAs, RNAs, such type of the molecules are separated due to the presence of different charge amount, due to the different size, shape, and therefore they move on to the cellulose, uh, polyacrylamide gel, electrophoresis, like in a case starch gel, agarose gel. Such type of gels can be used for the mobility of these biomolecules under the effect of electric field. And the LDH has more positive charge, and therefore it's fastest in the electrophoretic mobility. And in a sense that positively charged molecule will move towards a negative end, and like that. And therefore LDH has more positive charge, and it has been found that it moves fastly on the gel, while LDH is the slowest one. It possesses least positive charge. And here is the structure description of LDH isoenzymes, that what type of the units they consist of. LDH is an oligomeric. Oligomeric mean consisting of more than one subunits, and it consists of like four subunits. Therefore, it's tetrameric enzyme, and it's made up of four polypeptides subunits. It's an enzyme. It means that it must be consist of polypeptide sequences. And it has been shown that the two types of the subunits, which are namely M for the muscle and H for the heart, they are produced from the different genes. Because different genes are responsible for producing them, it means that when these subunits or the polypeptides units are uh, collectively they are present in a group, they will behave as the isoenzymes. Therefore, in the upcoming slides, when the M is written, therefore it will represent that these subunit is representing the muscle, whereas H will represent the heart. And M subunit is basic in characteristics properties, like basic in nature, while H unit is acidic one. And the isoenzyme, they contain either one or both of the subunit. Therefore, their combination gives the isoenzyme from LDH1, LDH2, LDH3, and so on up to LDH5. And the characteristic features of these LDH isoenzymes, they are given over here as like this table represents the different isoenzymes of LDH. And second one is the subunit constitutions, which are responsible for making these enzymes, isoenzymes. And the principal tissue or tissue of origin, that where they are originated and present. And next is the electrophoretic mobility, how they will move onto the electrophoretic bed or the gel. They will move fast, slow, what type of the behavior will be there. And whether destroyed by the heat, it means that denaturation, whether they will occur like at 60 degrees centigrade. And next is the percentage of the normal serum in human. What is their percentage in normal individual or in your normal healthy human being? Number first is LDH. It consists of the four subunits, and each subunit is H. Therefore, H is represented as the square, whereas M is represented as the oval or the circular shape. It means that in LDH1, all the four units they are H, and therefore it's written as H4. They are principally found, or their origin is from heart and red blood cells, and they move fastest on the electrophoretic in the electrophoresis. Their behavior is the fastest, and they have no effect like by elevating the temperature at 60 degrees centigrade, and normally their percentage in the serum is only 25%. It's low, but for the LDH2, it's H3M. It means that 4 is the H components for the heart, like 3 sorry, H3M means 3 components is for the heart. They are represented as squares, whereas the one component or the subunit is like M for the muscle. And although their behavior is not the fastest, but they can move very fastly on the in response to the electric field, and their percentage is like 35%. It is higher than the LDH in serum. And for LDH3, it means that they consist of two subunits for the heart and two subunits for the muscle. Therefore, it's the H2M2. And principal tissue of origin is brain and kidney. Uh, you can see from while moving from LDH1 to LDH5, their electrophoretic mobility goes from the fastest towards the slowest, and they are present in the 27%, and partially they, yes, they can be denatured or destroyed by the heat, and some extent some is recovered, like it's not affected by the heat, like temperature at 60 degrees centigrade. And for LDH4 and LDH5, they are present in the serum in very low concentration, like 8% and 5% respectively. But they are very heat sensitive, and yes, they can be destroyed by the high temperature. And their response is that as they have the basic nature, they move slowly on the electrophoretic gel, or their overall electrophoretic mobility is from the slow to the slowest. Next is, what's the significance of differential catalytic activity? Like what type of activity is affected if the levels of like LDH1, 2, and so on, if LDH5 is affected. Then significance lies in the proper functioning of like heart and the muscles. And here is shown that LDH1, which consists of the four H subunits, and it's predominantly found in the heart muscles, and it is inhibited by the substrate, which is the pyruvate. Like when the you can say glucose is converted into pyruvate, and pyruvate is the substrate which is present in the heart muscles, and pyruvate will behave as the inhibitor for the activity of LDH1, and hence the pyruvate is not converted to lactate in the cardiac muscles, but this pyruvate is converted to acetyl CoA, and this acetyl CoA will enters in the citric acid cycle, or the TCA, to give different entities, different compounds, and succinate, and so on, and oxaloacetate, and such type of the functionalities represents that yes, pyruvate will not be converted to the lactate in the heart muscles, but it will be converted to the acetyl CoA. Whereas for LDH5, which consists of all the four units as the M and therefore represented as M4, it is mostly present in the skeletal muscle, and the inhibition for of this enzyme by the pyruvate is minimal. It means that pyruvate substrate or inhibitor, pyruvate will not act as an inhibitor for the LDH5. It will not inhibit it. Therefore, pyruvate gets converted to the lactate. It means that lactate is going to be concentrate or accumulate in the skeletal muscles, and therefore LDH5 has a low Km value or it has the high affinity, while LDH1 has a high Km value, or we can say that it has a low affinity for the pyruvate. Therefore, when the LDH5 has the high affinity for the pyruvate, it will convert it into lactate, and LDH has the low affinity, and therefore this pyruvate will gets converted into the acetyl CoA. Next is the differential catalytic activities of the LDH1 and LDH5 in heart and skeletal muscles. It means that LDH1 and LDH5 have different catalytic activities in the heart and skeletal muscles, because LDH is important for the heart and LDH5 is important with respect to the skeletal muscles, and therefore they are well suited for the aerobic, like the presence of oxygen for LDH, and absence of oxygen like anaerobic conditions for the LDH5, like their presence in the skeletal muscles. And it's well evident for the conversion of like pyruvate into the like acetyl CoA in the muscle, while the pyruvate gets converted into lactate in the skeletal muscle, because they work under like in the anaerobic conditions, and therefore absence of oxygen, and there is differential or changing in the catalytic activities of the LDH1 and LDH5, which consist of H4 and M4 units respectively. Next is the diagnostic importance of LDH. What's their importance? What's the importance of LDH1, LDH2, and LDH5, so on? And isoenzyme of LDH have immense value for what? For the diagnosis of the heart and the liver and their related disorders. In healthy individual, it has been shown that the activity of LDH2 is higher than that of LDH1. When investigation has been done in the serum. In case of myocardial infarction, LDH1 is much greater than LDH2, and this happens when person has like a type of like heart attack, muscle contraction, muscle stress at the heart, and it happens within 12 to 24 hours after the myocardial infarction. And here is the diagrammatic representation of all these isoenzymes of LDH, and therefore you can see in the like first uh, up upper diagram, it shows the electrophoretic mobility and behavior of the lactate dehydrogenase, and like positive and the negative. It lies LDH1, it's more positive or it's more acidic, whereas LDH5 it's more basic one. And A pattern shows that the normal serum level Like you can see that LDH2 it's higher. LDH2 it's up, it's high, while in a case it's the healthy individual, there is much higher concentration of LDH2 and rest of them are low one. While in case of individual with myocardial infarction, LDH level is high. Also, LDH level is also gets high, but LDH1 precedes the LDH2. Therefore, it has high level, much high level for LDH1. While in a case when the patient has like the liver disorder, liver disease, then you can see that LDH5 level will be raised, whereas the rest of the level will nearly remains uh, unchanged. And therefore increased level of or the increased activity of LDH5 in the serum is an indicator of liver disease. And LDH activity in the red blood cell, it has been shown that it is 80 to 100 times more than that of in serum. And hence for the estimation of LDH or its coenzyme or its isoenzymes like LDH1, 2 and so on, LDH5, serum must be or the totally free from the hemolysis, or else false positive results will be obtained if because LDH activity is higher in the red blood cells, which is found that they are more in the red blood cell, like 80 to 100 times they are more than what's in their level in the serum. Therefore, they must be totally free from that. And next is the important isoenzyme, which is the Critten phosphokinase. And the Critten kinase or the Critten phosphokinase CK or CPK, they catalyze the interconversion of the phosphocreatine or called creatine phosphate to creatine. Like here is the representation: phosphocreatine gets converted to the creatine and creatine to the phosphocreatine. But they utilize the uh, like ATP or ADP. If it has to go in the forward direction, it means that ATP will be generated, phosphate group will be released. And it has been shown that CPK, it consists of like overall its existence in the body as three isoenzymes, which are CPK1, CPK2, and CPK3. But in comparison to LDH, which consists of four subunits, they consist of two subunits. Like CPK1, it consists of two subunits, which is BB. CPK2, it consists of two subunits. This is MB, and CPK3 consists of MM. And B is for brain, and M is for muscles. It means that the dimer which are composed of the subunits like stands for M, the muscle, and B for the brain. And the tissue of origin is like CPK1 brain, CPK2 heart, and CPK3, which consists of both the subunit as the M. Its main tissue of origin is the skeletal muscles. And in the healthy individual, it had been shown that the isoenzyme CPK2, which consists of MB subunits, it is almost undetectable in the serum. It means that CPK2 is present in a very small amount, very small concentration, which is only like it's the less than 2% of the total CPK levels. And but after the myocardial infarction, because they are present in the heart, MB mainly present, tissue of origin is heart, And therefore, within the first 6 to 18 hours, it has been shown that CPK2, its level rises, and increase in the serum, it's as high as like 20% against the normal, which is only 2%. Like in the healthy or the normal individual, its level was 2%, but in the myocardial infarction patient, its level rises to 20%. And the CPK2 isoenzyme is not elevated in the skeletal muscle disorder because its main tissue is the heart, therefore, it has nothing to do with the skeletal muscles. Therefore, we can say that the estimation of enzyme CPK2, which consists of subunits for muscles and brain MB, it is the earliest reliable indication of the myocardial infection that if its level rises, it means that the individual is not the healthy one. Its heart is going to be collapsed or it's affected by the stress factors, and therefore myocardial infarction will be there. And the words myocardial infarction is the heart attack, and it occurs when the blood flow decreases or stop to a part of heart, and therefore it causes damage to the heart muscles. It means that in myocardial infarction, heart muscles are badly affected. It's all about the isoenzymes, and you have gone through the two important enzymes, LDH and CPK, creatine phosphokinase. And for an assignment, you have to search that are there some type of studies or the literature is present which show that can you separate units of isoenzymes? If the answer is yes, then you have to brief the techniques which are used for the separation of these isoenzymes. It's all about today's lecture, and you can ask question or queries on the WhatsApp group in Google Classroom, and don't hesitate to ask any question. Best wishes, and Allah Hafiz, student.